Tonight I will be talking about why, the differences between immunity generated from COVID-19 vaccines and how they differ from that you get from an infection. To start with, I'd like to describe very briefly how the body fights infection, because that's important to set the stage for why we have taken the stance that we have. So, to understand the difference between immunity after infection and immunity after vaccination, we need to understand the way the body reacts to a foreign body. In this case, the SARS-CoV-2 virus. That's the virus that causes COVID-19. So when the virus enters the body, the cells known as the antigen presenting cells, APC for short, recognize it, they engulf it, break it down into its components. And these components are then expressed on the surface of the APC, where another part of the immune system, these cells are called T lymphocytes. They are activated by those cells presented on the APC, and they in turn activate all the soldiers in the body. So one of the soldiers is what is called the killer cells, and these are specific cells that target viruses or infected cells and kill them. And the other group of cells are called the B lymphocytes. These lymphocytes change into two types as well. One is a plasma cell. Those generate large amounts of antibodies that fight any virus that enters the body. And the other group of cells is the memory cells, and these lay dormant in the body, but can be reactivated by future infection. Okay, so the difference between immunity from an infection and immunity generated following vaccination is that instead of the whole virus being introduced to the body as you'll find in an infection, only one part of the virus is introduced. This means that with vaccination, there's a consistent reaction to one component of the virus. In this case, is the spike proteins from the mRNA vaccines that we use on island, and is given in. The advantage of vaccination is that the dose that is given to the individual is predictable, is measured and predictable. And there's immunity to a specific part of the virus, that's the spike protein, and you can easily measure it. With infection, the immunity to the virus can be to different parts of that virus, and the protection offered isn't consistent. The strength of the protection offered by vaccine is that it's designed to give the body the best possible chance of fighting off the virus with minimal side effects. The quality of the protection offered by infection depends on the amount of virus that enters the body to start with and how ill the patient becomes. So patients with milder infections generally tend to have lower level of antibodies in their system, whereas patients who have had severe infections generally resulting in hospitalizations will have a lot higher levels of antibodies in their systems. Vaccines are designed to provide protection over a wide range of mutations of the virus, as its target points on the spike protein are designed on the vaccine are quite broad. This is not always the case with natural infection. And the practical implication of this is that the vaccines provide a wider protection against variants of this virus and keeps outbreaks from starting. The length of immunity after infection varies very widely. As stated earlier, it depends on the strength of the infection and the parts of the virus that immunity is developed against. There have been studies to show that immunity following infection starts to wane from between 3 and 8 months. The immunity developed as a result of vaccines are much more consistent. Trial participants have evidence of ongoing immunity against the virus over a year after the trials have started. The total length of immunity following vaccination is still up for debate, as it is still being investigated. As I said earlier on, a year after the trial started, patients are still demonstrating strong immunity to this virus. The other point to make is that measuring protection offered after vaccination and natural infections can be quite complex. The commonly used test is the antibody test, which tests only one of the several components of immunity. As I mentioned earlier on in my statement, there are different components to this. It requires very careful interpretation as the level of antibodies fluctuates significantly and must be considered within the epidemiology context that we are looking at. So it's not just simple yes or no. So therefore, the national testing policy published by the COVID-19 Strategic Testing Committee reviews the evidence and states that antibody testing should only be used as part of patient or outbreak investigations. Finally, Bermuda, I'd like to remind you that several countries around the world have abandoned plans to achieve community immunity from infections. The costs in human lives, immediate and long-term ill health is simply too great. Many countries in Europe started out with this policy but have had to pivot because of the misery caused by deaths, poor outcomes and overwhelmed health systems. Recent data also shows that provide further documentation that even those who have recovered from COVID-19 infection still stand to benefit from getting vaccinated. The evidence continues to suggest that acquired immunity from vaccines still offers substantial protections against new variants now circulating across the globe. So, in closing, the quality of immunity that you get from vaccination is by far superior to what you get from a natural infection. Thank you.